students for this book. So I'd like to take 1 minute from all of you to show you how beautifully people have expressed their love for this book. Can we please have the testimonials running for the book? So the students from all around the country, they have given immense love to this book. Sir, I'd like to ask you, what was the gap in the field of microbiology where, you saw the need of this book? Why was this book needed for microbiology for
Yeah,
UG students?
yeah. Actually, see that today the students are preparing for the NEET UG and the NEET PG. NEET PG from the beginning of 11, 12, and NEET PG from the beginning of the first MBBS. See, what are the issue for such kind of the preparation? Basically, they are preparing for MCQ based preparation from the beginning. So what I am thinking is that they are actually losing to remember the long and the lengthy answer, like essay, full question, short note, around 5 mark, 10 mark. So how to overcome and how to keep the balance between these two, to focus and to remember lengthy long answer plus MCQ based preparation from the time of a UG. So to overcome all this, I had prepared is such a nice book, and also how this book is helpful to all you today. For that purpose, I had organized the webinar, and after the introduction of the CBME, competency based medical education curriculum, still there are certain issues that students are not actually grasping the real knowledge, or I can say the real principle of the microbiology. How to keep the balance between the CBME and also the microbiology subject, that also I am going to guide you people into the webinars.
Thank you, sir. So, microbiology is a vast subject, and all of us undergrad students, we need to learn how to frame our answers correctly and how to structure the answers so that we can get the maximum source. And that's what sir is going to tell us about. But before we go to the topic for today, let me take a minute to again introduce you to CBS Publishers. We are hosting this platform. We're hosting sir on this platform today, and I'd really like you to please note things about CBS PD. Can we please have the CBS PD PPT running? we are glad to have Modi sir with us. We'll be talking about how to frame our answers. Sir, we can begin with the presentation. After sir's lecture, I'll take up all your questions. So you can go on typing your questions in the Q&A box and we'll take it up in the end. Thank you. So we can start.
Yeah. Okay. PPT, please.
Yes, sir.
I will operate the PPT. It is better you give me the options. PPT.
So you have the share button on the screen. Share files and then you can run the PPT.
Yeah.
Third button from the left side, share files. Yes, sir.
Okay. See, as we all know, what is microbiology? It is nothing but the study of the microorganism and or disease produced by them. Now, what is the old microbiology? It was purely based on the study of a microbiology with a system like bacteriology, virology, mycology, parasitology, and organism study is then followed by the system-based study like a CVS infection, respiratory infection, GIT infection, like that. Now, what is CBME? In a CBME, it is the study of the infectious disease or system-based study, not or less the organism-based study. So, it is the infectious disease like a prevention and treatment, not the organism and its a diagnosis. Now, the problem with the CBME, actually there are lots of, there are minor problem with the CBME. See, as we all know, microbes, they do not have any specific anatomical border because one organism or its a species it can infect the multiple system. So, in addition, it is also not possible that we can change our laboratory of a microbiology department like a CVS lab, CNS lab, instead of the bacteriology, virology, mycology, or the parasitology lab. So, strict system-based study without the peer group study or without the clearance of the taxonomical concept, it may not hinder the real concept, but also put some erroneous perception in the mind of the students. Now, I'm giving one example how the CBME is little bit tough. Earlier, before the CBME, student they have a little bit of query. Suppose one organism like a salmonella. See, and this is the slide that is not prepared by myself, but in a one microbiologist group, many are sharing their view about the CBME. Little bit of issue with the students. Suppose one slide there that is kept. I kept it here as a one example. What one teacher is saying. Earlier, before the CBME, the students having a issue whether the salmonella it is gram positive or gram negative. But after the introduction of the CBME, the student is too much confused whether the salmonella is bacterium, virus, or the fungus or the helminth. So this is the major issue. Now, what is the solution? As we all know, microbiology is not the infectious disease. It is neither a therapeutic or the preventive branch. No doubt it is the clinical branch, but it is purely a diagnostic branch. We can't treat the patient directly, but we can advise the clinician to treat the disease. So there should be a balance between all this by describing all the systemic infection as a common infection, followed by the specific infection by the specific organisms. Now, like Example, suppose what is the meaning of the common infection, what is the meaning of the specific infection. Common infection like a caused by the multiple organisms like infective endocarditis, pneumonia, meningitis, and the specific infection that is by the particular organism like amoebiasis, that is by the entamoeba histolytica, gonorrhea, that is by Neisseria gonorrhoeae, tuberculosis, that is by the Mycobacterium tuberculosis, same way leptospirosis, candidiasis, malaria, filaria. So many, these are the specific infection. These are one example that how I had overcome all these issues. Suppose one chapter that is the bloodstream infection, common infection like a bacteremia, it is described at here. Then after one infection that is the pyrexia of unknown origin, that is described with all the related organisms. Same way infective anemia, and now in a below you can see the specific infection. So specific infection like the bacterial infection, salmonellosis, relapsing fever. Same with the viral infection like CME infection. So it is better to read like that, the common infection followed by the specific infection. And I had designed the book like that also. So ultimately, my book has been designed in a total 3 parts with a 11 section. Part 1 that covered the general microbiology, immunology, and the healthcare associated infection. Part 2 that covered the systemic microbiology from section 4 to 8. Part 3 covers the entomology, practical microbiology, and also the MCQs. This is the few pictures from the book part 1 with a different section. General microbiology section 1, section 2 immunology, section 3 healthcare associated infection, and now part 2 that is systemic microbiology, section 4 that is the infectious syndrome, or we can say the infectious disease. Then after section 5 that is the bacterial infections, viral infections, fungal infections, and the parasitic infection. And part 3 that also covered the entomology. See, as we all know, vectors, they have a too much role into the microbiology, and it covers one also the vertical integration with the community medicine. The section it is the entomology, and that is very nicely designed in my book. If you can read, you can realize. One more portion that is the practical microbiology. Even everything has been written into the book, but as we all know, students have to attend both theory as well as the practical exam. But practical microbiology portion, it is also part of the book, and that I had given. And as we all know, the third section of part 3 that is the MCQ, that also the core part of a study, because ultimately students are looking for the NEET PG good score. So it is better to start the MCQ based preparation from the time of a UG. So this is about the basics of our books. Now, why I had organized this webinar, as we all know, because after the NEET UG, students are habituated with the MCQ type of learning, and they are not able to remember the very lengthy or the descriptive answer. So how to keep the balance between the NEET PG preparation from the time of a UG along with the how to remember the lengthy and the descriptive answer. And second, to overcome the little bit of drawback from the CBME. Now, how to frame the answer into the general microbiology. Now, there are common topics of the general microbiology, like the important scientists Louis Pasteur and the Robert Koch. How to attain this type of answer? For that, there should be a common headings and subheading in your mind before you go for the answer. Like for the Louis Pasteur or the Robert Koch, these 4 headings should be in your mind, like birth to death, profession, contribution in the microbiology, and honor. Other common question from the general microbiology, like morphology of a bacteria, the topics like, or the question like short note, bacterial cell wall, bacterial capsule, bacterial flagella, or the spores. These are all headings that you have to cover under the answer of above mentioned questions. Which are the heading? Definition, antigenic property or the antigens, properties, example of the organism or the classification, biochemistry, function, and the demonstrations. Demonstration means diagnosis. Now, how to frame the answer from the immunological? Now, certain common question from the immunity, like innate immunity or the acquired immunity. If any question from or the short note from the innate immunity or the acquired immunity, These are the four things that should be part of your answer. Otherwise, I can say that the answer will be incomplete. Definition, properties, classification, mechanism, or the components. Now, other question from the immunology, immunoglobulin, like immunoglobulin G, immunoglobulin A, or immunoglobulin M. And these are the heading for the short note like IgG, IgA, or the IgM, properties, structures, subclasses, and the clinical significances. Serological test, they are also the part of a short note like Vidal test, VDRL test, and these are the headings that you have to cover. History, principle, antigen, antibody, method of the steps, result, interpretation, and lastly uses. Now the complement pathway, these are also the part of a short note like alternative pathway, classical pathway, lectin or the MBL pathway, and these are the headings as the answer. Definitions, properties, and the steps. Steps means flowchart. Now from the healthcare associated infection, still there are many questions which are common like healthcare associated infection including catheter associated UTI, ventilator associated pneumonia, that is VAP, SSI surgical site infection or the surgical wound infection, or the catheter associated bloodstream infection, and which are the common headings that you cover under these questions like synonym, definition, etiological agents, pathogenicity, laboratory diagnosis, prevention, and the treatment. Now other question like instrument, hot air oven, autoclave, they will be part of a certain they will be part of a short notes, and which are the headings that you have to cover like principles, instrument structures, method of use, article to be sterilized, holding time and temperature, advantages, disadvantages, and the sterilization control. Now how to frame the answer in the system. Now part 1 that is over. Now we are discussing about the part 2. From the part 2 the section 80, the systemic infection like common question, infective endocarditis, pneumonia, meningitis, gastroenteritis, and these are the subheadings that you have to cover like synonym, definition, etiological agent, pathogenicity, laboratory diagnosis, prevention. Now from the bacterial infection, these are the common heading, morphology of a bacteria, type according to the first. Before starting about the morphology, these are the headings. First, that is the type according to the gram stain with all the required figures. Then after shape and size, arrangement, capsule, motility, spore, other morphological feature like fimbria, L form, polar body, etc. If you keep all this heading and subheading in your mind at the time of the theory examinations, surely I can say that your answer is never remain unattended. If you have all this heading in your mind, then at least something will become in your mind to write into the answer sheet. So the morphology of bacteria, then after the culture characteristics. At first, start your answer with the effective factor, because to do the effective culture, all these parameters should be required, like a temperature of incubation, same way the pH of the medium, O2 CO2 effect, I mean the O2 aerobic incubation or the anaerobic incubation, and the other nutritional requirements that are part of the media. So first heading it is the effective factor with the 4 subheading like temperature, pH, O2 CO2 effect, and the other nutritional requirement. Then after the culture characteristics into the different media, followed by the automated culture like a maldito, Vitek, etc. And in addition to above 3 heading, in a few bacteria, also there are certain other cultures required like a culture into the animals, sometimes culture into the cells, and also the egg cultures. These are not for all, but these are for the few bacteria. Now the pathogenicity. Suppose there is a question, pathogenicity of a tuberculosis, pathogenicity of the pathogenicity of the Mycobacterium tuberculosis, pathogenicity of the Neisseria gonorrhoeae or the gonorrhea. Then you start your answer by attending all these headings in this order. The first heading, there should be a disease name like Neisseria gonorrhoeae causing gonorrhea, Mycobacterium tuberculosis causing tuberculosis. Then after other heading like a reservoir of the infections, source of the infections, mode of transmissions, incubation period, portal of entry, sites, precipitating factor, that cover 3: agent factor, host factor, and the environmental factor. Now this precipitating factor, they are part of the integrated study along with the community medicine, because all this again you have to read at that. So once you read at here, surely I can say it will it will save your time and also save your reading from the other subject if you read it from here. So precipitating factor, then after it is followed by the precipitating factor, then after it is the pathogenesis, clinical feature and the complication. As per my view, the last topic or the last heading under the short note pathogenicity of any bacterium, that is the complications. Now the lab diagnosis of the bacteria. As we all know, the lab diagnosis is not possible without the sample. And whenever such kind of the question asked like lab diagnosis of the bacteria, what student they are starting directly their answer with the different testing method like microscopy, culture, but they are not mentioning about the samples. Without sample, how it is possible to the diagnose? So start your answer about the lab diagnosis for the particular bacterium along with the samples. So first heading, that is the sample collection, and second heading, that is the testing method. And in the testing method, this is the order. First, it is the microscopy, that means the morphological properties of the bacterium. Then after the culture, then after the biochemical reactions, serological test, molecular method, and the other method like a specific test for the particular bacterium. Suppose Nagler reaction for the Clostridium perfringens, same way other particular test for the particular bacterium. And how to frame the answer into the viral infection. Now, how to start the morphology of the viruses? These are the headings: shape and size, genome and the capsids, envelope, papillomeres, and the others like an inclusion body, viral protein, site of the replications, multiplication, biosynthesis, all will be covered under the heading others into the morphology of the viruses. Again, I'm repeating, if you keep all these heading and subheading in your mind, surely I can say at least you have a certain things in your mind. So your answer is never remain unattained. At least you are able to write something if you have a idea about all these heading and the subheading into the examinations. Now the pathogenicity of the viruses. Again, these headings are almost same as like the bacterial infection. Start your answer with the first heading, that is the disease name, followed by reservoir of the infections, source of infection, mode of transmission, incubation period, portal of entry, sites, precipitating factors. Then after pathogenesis, clinical feature, and again the last heading under the pathogenicity, it is the complication. Same for the viruses as like bacterium. Now the laboratory diagnosis of the virus, again it is same as like the bacterium. As I told you, without sample you do not write the answer laboratory diagnosis. So your first heading under the lab diagnosis of the virus is the sample collection, and then after the different testing method, and this is the order: microscopy, culture for the virus. Routinely we are not using the nutritional media, but we can use the cell culture media or sometime maybe animal culture. Then after third heading, it is the serological test, molecular method, and the other test as per the specific virus. Now for the fungal, mostly fungals are described not as an agent but like a disease: mycetoma, candidiasis, aspergillosis, and these are the different headings under the any fungal infection like synonym, etiological agent, and the pathogenicity. Under the pathogenicity, the subheadings are reservoir of the infection, source of infections, mode of transmission, incubation period, portal of entry, sites, precipitating factor, pathogenesis, clinical feature, complication. Then after laboratory diagnosis, prevention, and the treatment. These are the different heading and subheading for the any fungal infection like candidiasis, mycetoma, aspergillosis, etc. Now and under the laboratory diagnosis of the fungal infection, even you can same as like the bacterium and the viruses, sample collection followed by the testing method, and testing method covers microscopy, culture, biochemical, serological, molecular, and the other method like a skin test, freeze mount, slide culture, cellophane tape method, hair bed technique, etc., including antifungal sensitivity testings. Now how to frame the parasitic infection. Now from the parasite, normally the question asked like the life cycle, pathogenicity, and the laboratory diagnosis. Ready? Now these are the different questions from the parasite. Now I am coming to the first topic, that is the life cycle. Now see how my book is totally unique with the life cycles of the parasites. That I will tell you. But before that, if there is any question about the life cycle for particular parasite, which are the heading that you have to cover? Your first heading, that is the host. Then after, method of a transformation. See, method of transformation, it is for a few protozoa, neither for the metazoa. It is not for the metazoa, but only for the few protozoa. Third heading, that is the method of a reproduction. It is only in all protozoa, but not into the metazoa. And the last heading, that is the cycle. I want to highlight one thing regarding the life cycle answer. See, life cycle, it is not possible without the host. Whenever we will ask any question about the life cycle, students are directly jumping over the cycle or the flow chart. They are not mentioning anything about the host. Without host life cycle, it is not possible. So what my request to all you is that always start the life cycle of a particular parasite answer with the host, like definitive host, intermediate host, like that. Without mentioning the host and directly jumping over the cycle, I think it is inappropriate or the incomplete answer. And your cycle, you can present in a two form, either in the form of the figure, you can present in the form of the figure and the flowchart. This should be part of your answer. And how I had make the life cycle and all over ultimately entire parasitology very interesting. See, as we all know, the life cycle of the parasite, whenever the students are reading the life cycle, at that time they are understanding each and every step. It is very easy to read, very easy to understand, but it is very tough to remember. Why? Because almost all the parasite life cycles, they are same. But once you close the book, what happened? It is very, again I'm telling you, it is very easy to read, it is very easy to understand, but very difficult to remember. Why? Because almost all the parasite life cycle are same, minor differences. So whenever you are reading, you feel confidence that you are knowing, but once you close the book, all the cycle will be together in your mind. So how to make the difference, how to remember all this life cycle appropriately? For that, I had tried little bit different. All the parasite life cycles are presented with a total 4 types of the color coding system, and I can confidently say that this color coding system will help you. It will improve your visual memory, it will improve your photogenic memory, and this will reflect whenever you are attending the answer into the exam. Now, how I make the color coding system? Total 4 color coding systems are given. One is the green color, that is the normal cycle. Normal cycle means, suppose one parasite enter into the human, it will come out. So the remaining part will be continue into the external environment. So normal cycle means cycle is continue between the human body and the external environment. That cycle is never going to be end. It is always continue. I am going to one more point, that is the traffic point signal. See, there are different traffic point. I had took this idea from the traffic point. In the traffic point, we are seeing red color signal, green color signal. What is the meaning of the green color signal? Green color signal means you can go. Same way, red color signal. Red color signal means you have to stop. I had applied this principle into the parasitology. Now how? As I told you, green color signal at the traffic point, that means you have to go. Here, green color into the parasitology means once the parasite enter into the body, it will come out and cycle is continue between the human body and the external environment. That's why all such life cycle at where cycle is continue between the human and the external environment, they are labeled with the green color yellow as well as the green color font into the figure and into the flowchart. This is the example I am giving you. Suppose one example, it is the entamoeba histological life cycle. You can see 2 color arrow, green color arrow with a green color font. Same way, red color arrow, red color font. What is red color? That I will tell you later. But at the moment, remember, green color arrow, green color font. That means the entamoeba histologica cyst enter into the body. Now it is coming out, cyst and the trophozoid. Now cyst and trophozoid, they will exit with the cyst into the stool, and again cyst will re-enter into the human body, and that will continue the cycle. So this is the green color because cycle is continuing between the human body and the external environment. Now I am coming to the second point, that is the red color. What is the red color? Red color means once the parasite enter into the body, it will not come out. That means human is considered as a dead end host. It's like a stop at a traffic point. If there is a red color signal, you have to stop. Same way, red color into the parasitic life cycle means parasite is entering into the body, but it is getting stopped. Human is act as a dead end host. Human does not allow that cycle to continue because human is not releasing that parasite external to the body or into the environment. See at here. Suppose echinococcus granulation cycle is continuing between the dog and the sheep, and this continuous cycle, it is labeled with the green color font as well as the green color arrow into the figure. Now you see the red color arrow. What does it mean? This red color arrow means suppose accidentally that egg, instead of entering into the sheep, if it enter into the human, then it will enter into the different organ. From that, it is not coming out because the human meat, it is not the part of her dog. To continue the cycle, it should be entered into the dog, but here it is not entering. Sheep meat is entering into the dog, but not the human meat. So ultimately what happened? Human is considered a dead end host. Human is stopping the cycle like a traffic point, stopping the further movement with a red color point or red color signal. Same way, red color signal means the cycle is getting stopped into the human body. And this green color and red color signal, surely I can say it will help you people to improve your visual memory. Whenever you are writing the answer, this color coding system, this photogenic memory, it will reflect in your mind. You will realize, okay, here there is a figure, the page has been changed, here there is a flowchart, here there is a green color arrow, green color font, here there is a red color arrow, red color font, and all this will help you to design your answer very perfectly without mixing one cycle from the other cycle. So these are the two color coding system. One is the green color, second it is the red color. Now third, that is the sky blue color. What it indicating? Sky blue color arrow or the sky blue color font, that indicating the auto infection. Auto infection means what? Here parasite is entering into the human body. It is also coming out. But what the difference it making compared to the red and the green? The difference it making is that before coming out, it will start one more cycle into the human body. Suppose I am giving you example at here. Suppose at here, one example of a life cycle, that is the MBBS vermicularis. Now what happened? The eggs are exit with the feces, and once they go over the external environment or over the soil, they will again re-enter by the injection into the human, and that will continue the green color cycle. Now, but sometime what happened? Whenever the female liberating the eggs into the perianal regions, what the female will do? It causing the perianal itching, and that enables the patient to scratch the perianal regions. And once the patient scratch the perianal region, what happens? The fingernail region, it will be contaminated, and eggs, instead of entering over the soil, it immediately, without visiting the soil, it is ingested by the swallowing the contaminated fingers. So ultimately, instead of entering over the soil or into the environment, eggs are entering or re-entering into the mouth. So this is the, before leaving the human body, it will start the new cycle. This is the auto infection, and same thing will be given in every life cycle where there is a auto infection cycle. And the fourth category of the cycle, that is the retrograde. Sorry, that is the black color arrow or the black color font. What does it mean? Hardly black colors arrow with a black colors font. It is mostly in one or two cycle. One is the MBBS vermicularis, second it is into the toxoplasma gondii. Now what it indicates? Black color arrow, black color font, it indicates the other cycle. Other cycle means other than above three: green, blue, and the red. Now the black. Now I am giving you an example. This is at here. See, you can see at here the black color arrow, it indicating the retrograde cycle. It is other than the auto infection, normal cycle, or the dead end cycle. Here what happened? The egg is hatching into the perianal region and liberate the larva, which re-enter into the intestine through anus. Ready? So without visiting the soil, again it is re-entering, and that is the retrograde. So all such cycle, one is the retrograde cycle into the enterobius ornicularis, and one more, that is the exoenteric cycle into the toxoplasma gondii. These two are the different cycle, and all these two are highlighted by the black color arrow as well as the black color font. So again I'm repeating, as like a traffic point, total. This concept, again I'm telling you, this concept I took from the traffic point at where green color, red color signals are available. Green color means go, same way green color into the parasitology means the cycle is continuing between the human body and the external environment. Second, red color at a traffic point means you have to stop. So what is the red color into the parasitology? Red color font, red color arrow means parasite is entering into the human body, but it is not coming out. Ultimately, human will be considered as a dead, and host human does not allow to continue the cycle. That is the red color. Third, that is the auto-infection. All such are mentioned with the sky blue color arrow as well as the font. It means before entering, before getting out from the human body or entering into the external environment, the parasite will start one more cycle, and that is the auto-infection. And finally, that is the other than above three are all mentioned with the black color arrows as well as the black color font. And surely I can say that this color coding system, this photogenic memory help all you students. Please follow this. It will improve your visual memory, and it also help you people how to differentiate one cycle from the other cycle without mix up. So it is about the life cycle of a parasite. And now the pathogenicity. Again, short note pathogenicity of any parasite, and these are the heading like a disease name, reservoir of infection, source of infection, mode of transmission, incubation period, exit form, infective form, portal of entry, same way pathogenesis, clinical feature, complication. Ready? In addition to the bacterial pathogenicity, viral pathogenicity, fungal pathogenicity, which are the other topic into the pathogenicity of the parasite, only two extra topic, otherwise almost same topic as like the earlier one. One is the exit form, second one it is the infective form of the parasite that you have to maintain, that you have to mention under the heading pathogenicity of any parasite short note. Right? And now the lab diagnosis, almost same topic. First topic that is the sample collection, and second topic that is the testing method. And testing method, these are the different tests like a blood sputum or the stool culture, then after the microscopic, particularly for the adult stage examinations into the specimen, followed by the microscopy, then after culture, it is culture that is less for the parasite, but for a few serological tests, then after molecular test, and the other method like radiological or maybe skin test. So this is about the all heading and subheading for the parasites. And as we all know, in a microbiology, you have to pass both theory as well as the practical. So again, I had covered all the practical aspect also in my 4th edition. So humble request all you to please follow. Surely I can say it will minimize your lots of hard work, and it will also help you people to prepare the practical along with the theory examination, and that is the practical microbiology. But practical microbiology doesn't mean all the questions are part of the practical examination. Few short notes are also coming from the practical topics, and which are like certain stains, KB method, ZN stain, they will be part of the short note. Maybe not routinely, but rarely. And these are the different headings that you have to cover, like KB method or the ZN method, history, principles, steps, action of the ingredients, result, and the quality controls. Ready? Now, certain media like selective media, enriched media, transport media, these are also the part. And my humble request, all you students, that please answer these media type questions by using the table rather than going for the descriptive presentation, because the table, first thing is that it is make your presentation very effective, and it will make the other impact in the mind of the examiner, and that enables you people to get even more marks. And once you go these type of the presentation repeatedly, you do not have to write down all these headings for a particular medium. Once you mention in a column, then I can say accordingly, then you can attend the answer for the different media. Now this is the table. The first row, that is the definition of a media like selective media, transport media, or the any other media which is asked into the examinations. And then in a second row, there are lots of column. The first column, that is the number 1, 2, 3, like that. Then after the name of the medium, suppose one example, that is the selective media, and you are taking one example, the Mekonki medium. So write down the name of the medium Mekonki and the other details about the Mekonki's medium, like important ingredients, action of the important ingredients, method of the sterilization, and the uses. So once you go with these type of the presentations, I mean by using the table for the different media, surely I can say repeatedly you do not have to write down all these heading mentioned into the second row, like number, name of the medium, important ingredients, action of the ingredients, sterilization method, and the uses. So it will save your time for the repeated writing. And now sometimes the culture method, they are also the short note, like make intros, field chart, or the aspect, and these all are the heading that you have to cover, like a principle, structure, catalyst, steps, advantages, disadvantages, and the controls. Now biochemical reaction, sometimes they are also the part of the short note, like immunities, catalyst, oxidase. Rarely we are asking, but even though these are also the choice of the certain examiners, and these are the heading that you have to cover, like principle, reagent, quality control, method, result, and the uses. So again, one thing is that, as I told you, which are the different heading and subheading required to attain the answer of a particular question. I had given the idea, but it is not absolute that these are only the heading and subheading. Sometime as per the organism or as per the question, also you have to skip some heading or subheading, and also you have to add heading and subheading which I haven't told you, and these are as per the different topic, and they are very nicely presented into the book. So thank you very much for the CBSPD for organizing such a nice webinar. All the students for giving your valuable time and participating into this webinar. Again, the participant and the organizing team. Thank you very much.
Thank you, sir. That was indeed insightful, and I hope all you students have taken your notes on how to structure your answers in microbiology. Now, I'd like to tell you a little about this book called Essentials of Medical Microbiology, authored by Dr. Prakash Modi. So this is the third edition of the book that we have, and the book is available on the Amazon platform, amazon.in, and on our website also on discounted rates. So you can go and purchase this book, because this book is strictly based on the new syllabus, on the new CBME pattern, and it is full of questions. It covers a total of 126 chapters. I hope, am I right, sir?
Yeah.
So this book is a complete coverage of medical microbiology for all you undergrad students.
One more thing.
And
sorry to stop you in between, but one more thing that I want to tell you. See, students are actually preparing for the NEET PG from the first year of the MBBS, and see what is the advantage in this book compared to the other. I'm taking one example. Suppose in anatomy, there are multiple books, like 2 to 3 volume of the gross anatomy, same way one book of the histology, one book of the embryology, and maybe other practical books like Cunningham and many more. So after reading all these UG's book, what the students have to do at the time of the NEET PG, they have to purchase one more NEET PG oriented book, which is MCQ based book actually. So purchasing all 8 to 9 books for the anatomy subject in a first year, reading all this book at least for 1 year, and whenever you have a NEET PG, you have to throw all this book and you have to purchase and you have to read a new book. What does it mean? It will increase your cost and also labor intensive work. Why? Because after reading 10 books, even in a first year, these are not help you people for the NEET PG. So for NEET PG, you have to buy the separate book. So what I had covered this in my book. In my book, more than 2,000 MCQs are given with the answer, explanation, and the mnemonic. And how it is useful? It is actually the MCQs are divided in unit total 3 category. Chapter based MCQ, they are given at the end of every chapter. What is the meaning of the chapter based MCQ? Chapter based MCQ means you have to require the knowledge of only one chapter to solve all the option of that MCQ of a chapter based MCQ. Now second category, that is the section based MCQ. Section based MCQ means whatever the options are given into the MCQ, for that you have to read entire section. Entire section means more than one chapter. And third category, that is the image based MCQ. So chapter based MCQ, image based MCQ, and the section based MCQ, all these three will help you people to take the grip of the entire chapter with a strong concept, with a strong principle. Now one more thing that I want to add, I was giving the example, that is the anatomy. After reading 8 to 10 books of a one anatomy subject, again you have to purchase the one more book, that is for the NEET PG. So ultimately, reading the new book at the time of a NEET PG, again it will take your more time. Also, it will increase your cost to purchase the one more book. What I had covered this disadvantage in my book. I had, as I told you, I had given three types of the MCQ: image based, chapter based, and the section based MCQ. Section based MCQ. So student do not have to take the other book, that is MCQ based book, for the NEET PG. If you have this book, it will cover entire your UG curriculum, practical microbiology, plus it will start your NEET PG preparation from the time of a microbiology UG time. So if you have this book, then what are the advantages? It will cover NEET PG preparation. You do not have to purchase the new book at the time of a NEET PG for the MCQ purpose. That will reduce your cost and ultimately burden of a multiple books. And the advantage, this book, if you read at a UG time, the read book, again, once you read after 4 to 5 years, it will not like a newer book. It will be very easy to read, to understand, and to collect the, and to rememorize all the concept of a UG that you read at the time of a second MBBS. So by many way, as I can say, parasitology portions really very interesting. Practical microbiology, that is also required, it is also given, and the NEET PG, all the MCQ. I think more than 2000 MCQ with a complete parasitology, complete practical microbiology, entomology, that is integrated with the community medicine, and entire UG. I think this is the unique book. As per my view, everything should have this book.
Thank
Thank
you,
you.
sir. So, because we are wrapping up right now, we have a few questions coming in from some students. So, I'll read
Yeah, please.
those questions, sir. We'd like you to answer, please. Nitya Sharma, she writes, how can we retain vast microbiology concepts effectively for exams? So, how to learn microbiology?
Yeah, as I told you, to learn the microbiology, the first thing is that go with a taxonomical concept, go with the peer group of study. Do not focus on the infectious based study, because as I told you, infectious based study will not give you the complete concept, or it will not clear your complete principle of the concept of the microbiology. Suppose I'm giving you an example. There are two organisms. One is the Neisseria gonorrhoeae and one is the Neisseria meningitis. Now, Neisseria gonorrhoeae, it is given with the genitourinary tract infection, and the Neisseria meningitis, it is given into the CNS system. So both organisms are actually having a same morphology. They are belong to the same taxonomical concept, and they are belong to the same clinical. I mean, both are the gram negative category. Both are belong to the same category. So if you go through this way, I mean Neisseria gonorrhoeae along with the Neisseria meningitis, peer group study, that then and then it will clear your concept far better, rather than reading these two organisms separately in a separate system. So first thing that you keep in your mind, that is the always keep the taxonomical peer group study in your mind whenever you are reading any organisms, because ultimately microbiology, it is the study of a organism, not actually the disease. And once you go for the disease, I think it will become infectious disease rather than a clinical microbiology. Thank you.
Thank you, sir. So, another question is from
What?
Shubh. What topics are repeatedly asked in the exams and we should never miss them?
Yeah, repeatedly. Actually, see, in an exam, it is depends on the examiner's choice. Few examiners are taking whenever they are setting the paper, they are taking 4 to 5 earlier exam papers, and they are organizing from that, and they are setting the paper. The important topic for a microbiology subject for the UG students are, I think, see, actually, it is in the microbiology we have two papers ready. So it is at a moment tough to say which are the important topic, but I can say from the general microbiology, like general properties of a bacteria, virus, fungus, and the parasite. From the immunology, antigen antibody reaction, antibody chapter, antigen chapter, complement chapters, complement system. I mean, these are the important chapter. From the healthcare associated infection, sterilization, and the disinfection. This is the most important. Same way from the bacteria, the important bacteria like Strepto, Mycobacterium tuberculosis, Mycobacterium leprae. Yeah, these are the important. From the viruses, the polio, influenza, and the coronavirus. These are the important. And most important from the virus, these are the hepatitis and the HIV/AIDS. These are the two. From the fungus, candidiasis it is the most important, and from the parasite, the entamoeba histolytica. Then after the malarial parasite, I mean the plasmodium. Then after the cystodes, trematode, and the nematode. These three groups. I think all these you have. If you have basic, if you did all these important topics, then I can say okay, at least it covered the 60% of all your important questions from the exams.
Thank you, sir. The next question is from Ansh. How to remember the lab diagnosis for all the organisms which are asked in microbiology?
Yeah, as I told you, the lab diagnosis, first thing is that always even see what the students. I am. This is my 23rd year into the medical science as a medical teacher, but always what I noticed whenever there is a questions into the exam about the lab diagnosis of the particular organism, students they are never mentioning about the samples. So without sample, it is not possible to diagnose any infection. So first you mention the samples and then go according to the different method. That is the testing method. That is the second heading as I told you in my PPT. And accordingly you can go for the order you keep in your mind. That is the microscopy followed by culture. Then after the biochemical, serological, molecular. This is the order, and lastly the other methods.
So, what is your message to the students for scoring maximum marks in microbiology theory?
See, the simple message is that as I had in a PPT, I had covered all the sections, all the important questions, and again I'm instructing to all the students, even requesting to all the students, please read the microbiology with a proper heading and subheading. If you have a idea about all the headings and subheading in your mind about particular question like Louis Pasteur, Robert Koch, pathogenicity of Ascherichia lumbricoides, pathogenicity of the Mycobacterium tuberculosis, or the lab diagnosis of the malaria, or the lab diagnosis of any other organism. If you have a idea about all this heading and subheading, first you mention all this heading into the answer sheet and leave at least few lines in between. Then try to remember. If you read something in your UG life, then I can say all this heading and subheading always help you people to write something without remaining unattained for the answer, because this heading and subheading it will give little bit stimulation in your mind. Okay, what I should write it here? Suppose I'm giving I am taking an example. There is a question about the laboratory diagnosis of the Neisseria gonorrhoeae. As I told you, first you mention sample collection, then the testing method. Testing method, the microscopy. Now try to remember. Microscopy means at least you have an idea. Now microscopy, which are the headings of the microscopy? These are the morphology, like a gram stain property. If you have an Neisseria gonorrhoeae, if you are not able to remember, even though your mind will say Neisseria gonorrhoeae it is the gram negative cocci, at least you are able to write down gram negative cocci word in your answer. But first thing is that the heading should be in your mind. So all this heading, it will help you people to recollect the memory. If you are, if intentionally you want to forget, then this heading does not allow you to forget any headings, any topics, any knowledge about it will give indirectly idea to you people. If I am missing, then this heading will help you. Something is writable at here. But heading and subheading should be in your mind before you appear into the examinations.
Thank you, sir. So we like to wrap it up here, and I'd like to show you once again. We come back to the love that we have received on this book. If you've not purchased your copies, you can go onto the website and purchase your copies. This book has been a wonderful support to so many students from different colleges. Everybody has given us so much love. Thank you, sir, for being a part of this episode on our platform, CBS Author Talks Live. We'll wrap it up here.
Okay, chalo. Thank you very much. Thank you.
Thank you, sir. Thank you, everybody.